Okay, so here we go, all about the cell and its parts. We definitely could say that cells are way too small to be seen by the naked eye, so you'd need a microscope. And the first thing that I would want to do is I would want to talk to you guys about the different types of microscopes that are available to use out there. Um, so this first part is about microscopes. One type of microscope is the basic light microscope. Which of course uses visible light. And passes it through a specimen. And of course there's magnification. The way that you determine the magnification, if you think about a microscope, there's actually two lenses. There's a lens on top and then there's a lens on the bottom. This is where your eyepiece is at. This is where your actual larger lens is at. And to find total magnification, you have to multiply the two. So if the eyepiece, the eyepiece is typically about a times 10, and then the different rotating lenses here could be anywhere from 10 to 40. So if it was 40, I would take 40 times 10, which is what? 400. So the light microscope is one type, and it's good, but the problem is that it really can't magnify too much, because if you think about it, you could only magnify as far as light can resolve, and the wavelength of light is approximately like 450 or so nanometers on an average, which means that there's like a limitation on how big objects could possibly be, or how, better put, small objects could possibly be before they're unresolvable. So the wavelength of light, how quickly the light waves oscillate essentially, could prevent you from seeing something very small with a light microscope. And so you would need something a little bit more intense. And so when we look at things more intense, we would probably want to use like some other type of microscope, probably an electron microscope. Now when we focus on our electron microscope, we can see that there are two types that we would want to discuss. The first type is something called a scanning electron microscope. And the second type is the transmission electron microscope. The scanning electron microscope is really good for seeing the surface of objects. It has the potential to provide 3D images, whereas the transmission electron microscope is able to focus a beam through light. And focus a beam of electrons through a specimen, which means that it's going to go straight through the specimen.
So would you see like the insides of it?
You can see like different layers of the insides of it.
Okay.
So you see, yeah, I mean essentially you can see the insides of it.
So if like you were looking at a cell, you'd be able to see all the
Organelles inside of it. So I've got a picture I can show you.
Oh, your computer's working.
They fixed it.
Well, I missed a lot of them yesterday. Computer's fixed.
My life is fixed.
Okay. That works.
It's all better.
Yeah.
So anyway, you can see there's the SEM, that scanning electron microscope. Hopefully you can see through this that like I'm seeing the outside of the cell, and I'm getting like a pretty three-dimensional. See, there's like depth, if you will, in that picture, as opposed to the transmission electron microscope where you don't really see anything on the outside like you did here, but you can see it to the interior, perhaps some of the organelles or cross sections of objects. Cool.
What organism is this that we're looking at? Just like a bacteria?
I couldn't say. Very possibly. Cool. Now let's talk about organelles. What are organelles?
The organs of the cell.
Okay, so these are the tiny organs of a cell.
What about the less?
Bless you. And these functional units of a cell may or may not have a membrane. But we could agree that they are non-living, which means they can only function as a collective, or they function best as a collective. Cool. Now, one thing that we do want to focus on, though, is we want to focus on maybe how important it would be to isolate the different organelles from one from another. And that would involve a process called cell fractionation. So let's talk a little bit about cell fractionation. Again, that's a process that can separate organelles from each other. So you can separate organelles from each other. Of course, when you separate them out, you obviously can study them independently, which would be a huge advantage. Even though, as we had stated before, they probably won't function as best, at least they still would function, and you could study maybe their biochemistry, or something else, some other pivotal or important piece about that specific item. And so let me show you what you do real quick. And it's kind of best that I show you with a picture. Let me get a picture. Awesome. What you can see in this picture, and this again is in your book as well, is you'll notice that at the very beginning you're starting out with like all these different like tissue cells, correct? And you homogenize them, which means you basically cut open the cells, and now the cell contents are everywhere in this mixture. So you don't have like a little container of cells, a little test tube of cells. You have like a test tube of just cell components. You're seeing right
So
through
are
there.
you just legitimately blending it?
Yeah, you're essentially, yes. And then you put it in a centrifuge. And what a centrifuge does is it's an object where it just spins around very fast and a lot. And what you have is, each time you do that, settling at the bottom is something that's typically very heavy. One of the first things you would have is a nucleus. It's one of the heaviest parts of a cell. So if you remove the nucleus, remove this bottom layer, it goes away. You can centrifuge it again, and you're left with mitochondria, and maybe chloroplasts if it's a plant cell or a protist. The reason you have mitochondria and mitochondria and chloroplast this time is because they're both pretty large. You could extract them. That means that your liquid at that time, right through here, would be containing like only pieces of membrane, maybe some ribosomes, some other small organelles. And you see how I can fractionate, which means like I can piece by piece take these components and separate them out. So if I want to study the chloroplast, I have a technique for removing chloroplasts. The same thing goes with mitochondria. Cool. Awesome. It might help you on your paper if you write like C picture. I don't know. Okay, here we go. Okay, eukaryote and prokaryote. About prokaryotic cells, we know that they have no nucleus. The eukaryotic cells do have a nucleus. Talk to me about organelles. Do prokaryotes have organelles?
Not membrane-bound ones.
That's the answer I'm looking for. So yes, organelles, I'm going to write for prokaryotes. But each of the organelles must lack, or does lack, a membrane, which means that their organelles don't contain like an outer barrier. Their organelles are just located pieces of matter. Eukaryotes do have organelles, and a lot of times their organelles do have a membrane, which is pretty cool too. And you can see the obvious trend here as you go from being lesser evolved to more evolved, from prokaryote to eukaryote. The prokaryotes evolved approximately 3.5 billion years ago, whereas the eukaryotes evolved something like 1.8 billion years ago. So there's an obvious difference like in time between the two. What kingdoms out of my major kingdoms would I put under the prokaryotic group? Archaea would be one, and eubacteria, sometimes just called the bacteria. Your 6 kingdoms that I would put under the eukaryote would be, or your 4, I'm sorry, Protista, fungi, plant, and Animalia. Do it? What about it? A protist is a single-celled creature, typically single-celled, and it's going to be probably living in an aquatic environment. And they are eukaryotic because they have membrane-bound organelles, but that kingdom has actually been subdivided into like 27 kingdoms currently. So like it went from being the random, like I hate to, this sounds weird, but like the random junk pile, if you will, where like it was just a little bit of everything, and they've organized it in the last 10 years or so. And their organization took them from a 7 or 6 kingdom system like you're seeing here to like a 35 to 40 kingdom system. And that's mostly because they took the protist phylums, which is the level below kingdom, and they made all those phylums kingdoms because they were unique or distinct enough that they couldn't be all included in the same protist kingdom. So this term today is kind of like fizzled away, and it's just like a journal term that describes the 27 kingdoms that used to be all phylums of the Protista kingdom. So no, we won't learn all. We will not learn any of them. In fact, we won't spend a lot of time with them, just because it is a lot. when you look at both the prokaryote and eukaryote, you could understand that it would be important for us to recognize that there's got to be something common between them. There's got to be a lot of common features. And so let's go through some of those features that are common between the two groups, if you will. We'll write those down real quick. We'll describe what they do. Okay. As far as the plasma membrane goes, what's its function? What is it? What's its function?
It really facilitates osmosis or whatever the other one is.
Okay, so it's an outer barrier, right? And it can facilitate or allow maybe entry or exit, depending on what we're talking about. What's it's made of? What is it made of typically? Two things. That's one of them. What else? That's another good one. Not the primary one though. Phospholipids is the primary one, and then proteins is the other one. Now cholesterol is found in there, but it's found in a very small percentage, and carbohydrates are too, but in a very small percentage. Your bulk is it's 65% phospholipid, 35% protein, and it's actually 34.9, and it's actually 64.5 or whatever, and the other 0.9 of everything is the carbs and the cholesterol. What about the cytosol? The prefix cyto means cell. Do we know what the cytosol is? It's the internal fluid of a cell.
So it's the same, we learned it as cytoplasm.
Mm-hmm. Cytoplasm, though, is referring to a general region. Cytoplasm is everything that is between the nucleus and the membrane, so it's basically everything in the cell besides the nucleus.
Okay.
The cytosol is the fluid that you associate with being cytoplasm. Cytoplasm, in your mind, might seem as though it's a fluid that's very gel-like. That's true. No, not really. Cytoplasm is the area that contains the cytosol.
Okay.
The word cytosol and the gel or liquid that you've got in your head, those terms go together. Cytoplasm contains the cytosol. Chromosomes, how about this? What is their function? What are they? Okay, so they carry the genes in distinct units. And how about ribosomes? What's their overall function? Perfect. I almost misspelled the word synthesis. So protein synthesis. Now let's talk real quickly, going back up to prokaryote, because we'd said prokaryotes didn't have a nucleus. Do you know what their solution is to their no nucleus situation? Since they don't have a membrane-bound container of chromosomes, speaking of chromosomes and ribosomes, they have something called a nucleoid region, which is an area where the DNA is found, the chromosome, because they typically have one chromosome, where their one chromosome is found, but it's not a membrane bound. So we could write under this, no membrane. Cool. Awesome. Why are your cells small? What I'm gonna get you to do real quick is to take a couple of seconds, and with someone around you, I want you guys to think about why cells would have to be small. This is actually a really important topic, so think diligently.